BNR biomasses under microbial communities are often considered as black box. Let's open them using conceptual ecosystem models. I will show you how you can connect the dots between the metabolisms of different guilds and microbial populations of a BNR microbiome. This will lead you to develop an ecosystem model. We will further discuss the use of ecosystem models in PNA, EBPR, and BNR systems operated with activated sludge and granular sludge or biofilms. You know how to draw simple sketches of metabolisms of microbial guilds. An example from the textbook figure 2.10 is given here for a partial nitritation and anamox ecosystem. On the panel A, simple metabolic sketches are given for nitrifiers and anamox bacteria, as well as competing denitrifiers. To do these schemes, you need to list upfront the electron donor couple, the electron acceptor couple used in their catabolism, the carbon source and nitrogen source assimilated by anabolism. You do this for every guild. Then you connect the dots by looking at which substrate is used by different populations and which product is produced by one organism and used by a next organism. It leads in the panel B to the progressive development of the microbial ecosystem and metabolic network of the main metabolizing guilds. Besides them, there can be several flanking populations that are present in the microbiome. We do not know exactly their function in the microbiome. Some populations are presumed to thrive on lysis product from the main metabolizers. I would like that we develop now an ecosystem model based on a previous exercise that we did on the growth systems of BNR microorganisms. Please take a sheet of paper or a tablet and first sketch the individual metabolisms of these BNR guilds based on the information compiled in this table. Then connect the dots to build up the conceptual ecosystem model of the microorganisms and microbiome. You can leave out these fermenters first for this exercise. You can pause your video. Answers and examples will be given in the next slides. Here you can find the metabolic sketches of most of the BNR guilds of heterotrophs, nitrifiers, including the Comamox, denitrifiers, and anamox organisms. I then connected the individual metabolic sketches to reconstitute stepwise a conceptual ecosystem model for this theoretical biomass. The BNR guilds interact in different ways in this theoretical network. Organic matter is used as an electron donor and carbon source by heterotrophs and denitrifiers. Both guilds are chemo-organo-heterotrophs producing CO2 or bicarbonate at pH 7. OHOs respire aerobically using oxygen as an electron acceptor. DHOs respire anaerobically using oxidized nitrogens like nitrate or nitrite as electron acceptors. OHOs use ammonium as nitrogen source. In this simplified example, DHOs use nitrate NO3- as nitrogen source, considering that ammonium gets fully nitrified by nitrifiers. So nitrite and nitrate are produced by the oxidation of ammonium by ammonium oxidizers and nitrite oxidizers. Comamox organisms in light blue can potentially coexist depending on conditions by oxidizing ammonium aerobically up to nitrate. If anaerobic conditions are established in the system, such as in a biofilm reactor, anamox bacteria in purple here can use ammonium as an electron donor and nitrite as an electron acceptor. Nitrifiers and anamox organisms are chemolitho-autotrophs and use the CO2 or the bicarbonate produced by heterotrophs and denitrifiers as carbon source. Besides catabolisms, the carbon and nitrogen resources are assimilated in all these microbial biomasses through anabolism. This exercise gave you some first insight in the development of conceptual models of a BNR ecosystem. Another illustration is given here by Per Halkier Nielsen and colleagues on the development and use of a conceptual ecosystem model to predict the performance of eBPR. The left panel shows the different inputs and outputs from the process, the system analysis, and the typical process control tools that you can apply to manage the process. It shows also the microbial analysis that can be conducted for better knowledge on microbial populations and their ecophysiologies. The ecosystem model leads to a system's microbiology understanding of the sludge, in parallel to the improvement of the treatment plant design, operation, and troubleshooting. In the center, the frequently abundant phylotypes that populate eBPR sludge are given together with their metabolic traits that can be derived from annotated MAGs, metagenome assembled genomes, or verified by ecophysiology analyses like SIP or microautoradiography. On the right, you can see under which conditions examples of nitrifiers like Nitrosomonas, denitrifiers like Tauera, fermenters like Streptococcus, PAOs like Candidatus acuminibacter, as well as filamentous bacteria like microtrix that hydrolyze particulate organic matter are selected along redox conditions in this flow-through system. I am giving you here a final illustration on the ecosystem model of BNR granular sludge, also known as aerobic granular sludge. I built this model based on the microbial populations detected in the ecosystem and the knowledge that I gained on their metabolisms. Let's follow the arrows. Depending on the type and geometries of the sewer system, organic matter can enter this granular sludge as particulate organic matter, soluble organic matter, mainly XS or SS respectively, or as dissolved volatile fatty acids. These VFAs result from the fermentation in the sewer system. Short and steep sewers often result in a relatively high amount of XS in the influent. Long and flat sewers are more prone to fermentation, resulting in more VFAs in the inlet. During the anaerobic feeding phase of the granular sludge system, XS, SS, and VFAs are subjected to hydrolysis, fermentation, and storage, respectively. Different microbial populations are involved in this metabolic sequence. PAOs and GAOs can take up the VFAs as polyhydroxyalkanoates. When aeration is switched on, PAOs and GAOs can grow on the PHAs that they have stored as carbon source and electron donor. The diversity of PAOs and GAOs is under continuous investigation. We call a PAO and GAO saga. Organisms like Candidatus acuminibacter, Dechloromonas, and Tetrasphera are known as PAOs. Their exact contribution to phosphorus removal remains to be quantified. Tetrasphera has a fermentative metabolism to ferment organics. It's a bit different than the metabolism of Acuminibacter. Under aerobic conditions, nitrifiers can grow by oxidizing the ammonium into nitrite and nitrate. These oxidized nitrogens can diffuse further in the granule to get used by denitrifiers as electron acceptors, eventually producing N2. A wide diversity of denitrifiers, or DHOs here, have been detected from BNR granular sludge. However, it remains puzzling which electron donor and carbon source they use if not able to store it anaerobically as PHAs. Denitrifying PAOs and GAOs are generally conceived to simultaneously remove phosphorus and nitrogen, or maybe their diversity is much more widespread than generally thought. If given the chance, anamox microorganisms can develop in the anaerobic biovolume of granules, but they are much more slower growing than denitrifiers. One way has been to inoculate BNR granular sludge with anamox granules, which get covered by other BNR organisms. BNR granules can be conceptualized as an ecosystem of EPS producers and consumers in the biofilm matrix. In another recent study, we examined with Sergio Tomás Martínez and Teudelf colleagues the EPS turnover through biosynthesis and biodegradation cycles in a substrate labeling experiment using 13-syn acetate. The results indicated that EPS from the PAO-enriched community was not selectively degraded by flanking populations per se under stable EBPR process conditions. Instead, a general decay of biomass was observed, resulting in the release of lysis product that can be reused by other populations. In conclusion, conceptual ecosystem models are nice to aggregate taxonomic and metabolic knowledge about microorganisms in microbiomes of wastewater treatment plants. It helps crack the network complexity. Ecosystem models provide a strong basis for questioning BNR microbiomes and designing functional analysis, such as via ecophysiology and substrate labeling. Activated sludge and granular sludge are model ecosystems for the application and testing of microbial ecology and microbiome engineering principles.